Good morning all. So I'm going to present the data from proteomic analysis of limited number of cells using adaptive focus acoustic technology. So we are at the mass spec conference, and we all know that with the current generation of mass spec, proteome analysis even at the single cell level is feasible. However, as easy as it sounds, it is quite challenging and difficult, especially in terms of sample preparation strategies. So when we start with low starting material, the multi-step sample processing workflow that we generally use for preparation of sample for proteomic analysis, it can cause sample loss leading to reduced protein recovery and limited protein identification. And there are various biological samples where we may be limited by the starting amount of material, and the proteome analysis of these samples is of clinical value, such as biopsy samples or cells enriched for certain population of cells like immune cells enriched from whole blood cells, etc. So this is a conventional bottom of proteomics workflow for analysis of cellular proteome. So the first step is cell lysis for extraction of protein and solubilization of protein, and there are various methods of choice to accomplish these steps. There are physical methods as well as chemical methods, which includes addition of detergent and salts. However, if such methods are performed in a non-controlled environment, they can be non-reproducible, and addition of detergent and salts require an extra step of cleanup, as these may not be compatible with the mass spec. Once the proteins are solubilized, reduction and alkylation happens, and followed by digestion, which is usually an overnight step. And as I have mentioned, cleanup is required before the sample can be analyzed on mass spec. Now this is a very general workflow with choices of options available at each step, creating many variations, and there is no universal method adopted as such. SP3 method is one popular method which stands for single pot solid phase enhanced sample preparation. This is essentially a cleanup substitute. So once the proteins are solubilized, reduced and alkylated, paramagnetic carboxylate coated beads are added to the sample, and there occurs a non-specific binding of proteins to the beads, and these beads, they trap the protein in a hydrophilic layer when there is a high organic solvent present in the sample, which facilitates the washing of the protein bounds to the beads. So in the presence of an organic solvent like ethanol, the salts or the detergents or other contaminants which were introduced during the upstream processing can be washed away, and finally the bound protein can be eluted in an aqueous environment under the presence of a magnetic field. Or we can choose to digest the bound protein on bead, and the digested peptides can be collected, and these peptides are ready to be analyzed on mass spec. And because this is this uses magnetic bead, this method is easily adapted on automation, making this a high throughput method. And we are using this method for a number of different sample types like FFP, cell lines, patient samples, etc. And this has been shown to be reproducible, especially at the lower amount of protein. So the AFA assisted protocol also uses magnetic beads and on-bead digestion. In addition to that, the complementary addition here is the use of AFA technology at each step of cell lysis, binding of protein to the beads, and during the digestion. And as Deb has mentioned about the True Prep kit, this kit provides all the reagents and the accessories required to accomplish this protocol, and it is the protocol is compatible with the R230 focused ultrasonicator platform. So one thing to consider about the AFA technology, that the high energy ultra frequency which is applied to the sample, it is happening inside this platform, which maintains an isothermal and non-contact environment. And when the ultra high frequency energy is applied to the sample, it creates the hydrodynamic shear, which is adding an advantage at each step of cell lysis, binding and digestion. So our objective here was to compare the standard SP3 digestion protocol that we were following to the AFA assisted protocol, and also to see that how the AFA assisted protocol is performing across a range of cells starting from 1000 cells. So to compare the two protocols, we took HUCCT1 cell lines, which is a cholangiocarcinoma cell line, and we took 500,000 cells and 100,000 cells in triplicate in two different sets, and one set was processed with the SP3 method and another set was processed with the AFA assisted method. So there are few differences in the two methods. In the SP3 method, during the sonication, we had to add benzonase for the shearing of the nucleic acid material, which was not required in the AFA assisted method because the AFA technology is taking care of that. And during the digestion, in the presence of AFA, the digestion is happening in an hour, while in the SP3 method, it is usually an overnight protocol with agitation on the thermomixer. So once the peptides are generated, the data was acquired on a thermo Orbitrap SN over a 120-minute long run. And these are the bar diagrams showing the performance of the two methods. So in terms of protein identification, close to 4,000 proteins were identified with both the methods at both the cell numbers, 500,000 cells and 100,000 cells. And both the methods were reproducible across the different replicates as represented by the error bars. And the similar results were similar results were observed at the peptide numbers as well, although there were a slightly higher peptide count with the AFA method, but the difference between the two was not statistically significant. And these are the Venn diagrams showing the overlap of the protein identified with the two methods across the three replicates, and these shows a good overlap. We also compared the length of the peptides that is being generated during the two digestion method, and we found that these were similar across the two methods and the replicates, and most of the peptides generated were between 8 to 15 amino acid long residues. We also compared the miscleavages along the two methods, and most of the peptide almost 90% peptides present were with no miscleavages, and there were less than 1% peptides which were identified with two miscleavages. However, the difference between SP3 and AFA assisted method was non-significant. And we also compared some of the common modification that we usually use during the database searches for our data, which is a deamidation of asparagine residue or oxidation of methionine residues. The difference between two methods that we found were not significant. So with this we conclude that AFA assisted method shows similar performance as compared to the SP3 method. Next, we wanted to see the performance of AFA assisted method across a range of cells starting from 1000 cells. So we took 1000 cells, 10,000 cells, 100,000 cells and 500,000 cells and processed them using the AFA assisted protocol and analyzed on mass spec in the similar manner. And 1000 cells equivalent was injected on the mass spec for analysis. And in terms of number of proteins and peptide identified, we found similar results across the range of cells. Close to 4,000 proteins were identified at the 4 cell levels, and these were reproducible across the replicates, and similar observations were made even at the peptide level. In terms of peptide length as well, AFA assisted method showed consistent performance across the cell numbers, and there was no significant difference in terms of miscleavages as well. 90% of the peptides identified were with no miscleavage at all, and similar observations were made when we compared the modifications. So with this we conclude that AFA assisted method also perform consistently when we are comparing a range of starting amount of cell numbers starting from 1000 cells. However, one big difference between these two methods is that the sp3 method follows an overnight digestion, making it a longer method. However, in AFA assisted method, digestion is accomplished in 1 hour, so reducing the turnaround time. So finally, we concluded that AFA assisted method shows similar performance to sp3 method, but with reduced turnaround time. And because of the use of magnetic beads, this method also has potential for automation, converting it into a high throughput method. And I would like to thank people from my research group as well as Covaris team. And this is our research group at Mayo Clinic. Thank you.